So I'm going to talk to you today about some predictors of cognitive performance that we've established from the Aberdeen birth cohort data, and hopefully also show you how these findings link in with some disease data from a disease cohort of ALS patients, and how this might be relevant to precision medicine. Sorry, there we go. So just start off to introduce myself a bit, because I haven't been to the Health Data Sciences Centre before. I started in Aberdeen with my PhD with Dr. Gordon Waiter, who some of you might have met from Aberdeen Biomedical Imaging Centre. I think he's here online as well. And Chris McNeil, who's also in the Biomedical Imaging Centre, working alongside Alan Snedden to look in this Aberdeen children of the 1950s birth cohort at brain iron, which I'll come back to a bit later on in the presentation, and how it's related to age-related cognitive decline. And in 2023, I finished my PhD and moved to the Gregory lab in the IMS, where I'm looking at molecular crowding and how that relates to amyotrophic lateral sclerosis, which is ALS, which is a neurodegenerative disease. So my main research focuses in this role is looking at precision prevention strategies in neurodegenerative disease. So just a bit about what I mean by these precision prevention strategies. So if we're looking to intervene in patients who are symptomatic, then by the time this person's presenting at clinic with symptoms, they've already undergone a lot of irreversible cell death, a lot of pathology. Whereas what we're aiming to look at with precision prevention strategies is to look a bit earlier in this asymptomatic group, which has pathology but isn't presenting with symptoms. And so we might be able to detect these signs of pathology then, in order to treat the right person at the right time with the right treatment, and therefore prevent the disease starting, or at least reduce the disease burden. And so the idea of this is to look for earlier detection mechanisms for the disease, and then stratify these people by what different disease subtypes they might have, in order to provide an earlier and more targeted intervention for better outcomes for patients, and potentially to prevent disease, which is the ultimate goal. Today I'm going to talk about 3 different cohorts that we've looked at. Firstly, I'll talk about an asymptomatic cohort, which spans across this group that has no clinical signs. And then I'll talk about a purely symptomatic cohort, which is looking at ALS patients who are already presenting with clinical signs. And then I'll talk about a cohort that we call the at-risk cohort, where we're looking at people who have pathology present who don't have a diagnosed disease. So cohort 1, I'll just briefly introduce these, and then I'll talk a bit more in detail about them later on. Cohort 1 was the Aberdeen children of the 1950s birth cohort and their first-generation relatives, who do not have any self-reported neurodegenerative disorders. In this cohort, we wanted to ask which measures are predictive of cognitive performance. In cohort 2, we looked at a symptomatic cohort of post-mortem brain tissue from ALS patients. We wanted to ask here whether predictors from cohort 1 were associated with pathology that we're seeing in a symptomatic cohort. And then following on from that, we looked at a third cohort of post-mortem tissue from people who have ALS, but also who have Alzheimer's disease and some non-disease control cases, and to see whether we can identify these pathologies outside of disease. So firstly, let's talk about the asymptomatic cohort. And a lot of you will be familiar here with the Aberdeen birth cohorts, which we're really lucky to have access to at the University of Aberdeen. And the straddle study included not only the Aberdeen children of the 1950s, but also their first generation relatives. So it gives us a bit more of a span in age range. And all of these people that we included in the study had no self-reported neurodegenerative disorders. So there was quite a lot of phenotyping of this cohort. We initially had access to data on risk factors like age and weight and sex and Scottish Deprivation Index. We also had full blood counts available, and as part of my PhD, I started to do some more targeted analysis of the blood tests that we had, looking at specific ELISAs for iron status and inflammatory status, which was what my interest was at the time. And we also worked with Gary Cameron in core facilities to do some ICP-MS and measure the heavy metal levels in the blood samples of these participants. Also during my PhD with Gordon Waiter, we looked at iron deposition in the brain and volumetric assessment of brain regions, and we used a susceptibility weighted imaging technique to directly measure this iron deposition. And then with Hamish Johnson from the School of Geosciences, I was able to look at atmospheric pollution exposure indicators based on the participants' postcode information. And we, on the same day of blood testing and brain imaging, these participants all underwent several cognitive tests, looking at very different aspects of cognition, including memory, processing speed, verbal fluency, for example. And we combined all of these different cognitive assessment scores into one general cognition score, or G score, which I'll refer to throughout the presentation. And this encompassed all 6 of these using a principal component analysis. So to actually try and identify potential predictors of cognitive performance, we used a random forest classification technique. We were really interested in identifying the group with the poorest cognitive performance levels. So this would be people who would potentially be at risk of going on to develop neurodegenerative diseases. And to do this, we used this random forest classification where we split the participants into tertiles based on their G score, and we trained the model on 80% of the data, and we then tested the model on the remaining 20% of the data. So just to clarify, going forward, the tertiles that we split them into were the highest cognitive performers, the lowest cognitive performers, and then this middling group, middle performing group. And actually overall, our model predicted the purest cognitive performing group with quite high sensitivity and specificity, over 90% specificity and over 70% sensitivity. So we were quite confident in the model, and we went on to perform a leave one out analysis to identify potential predictors, where we took a variable out of the model and saw whether the model predicted what class of cognitive performance people went into, better without the variable or better with the variable. So these variables out of the 457 that we put in came out as the greatest predictors of cognitive performance. And I'll go into each one in a bit more detail in the following slides, but as you can see, there was a small number for each group, with quite a lot of environmental pollutants coming out as predictors. So first off, we looked at the potential risk factors that we had sort of expected to come out as risk factors for cognitive performance, such as age and education. And also we included the Scottish Index of Multiple Deprivation, but actually out of around 44 variables in that category, only 3 came out as predictive of cognitive performance in this model. And weight but not BMI were predictive of cognitive performance, which I guess is interesting to look at BMI as a measure. Next, we looked at the blood tests, and we found that routine blood tests actually predicted cognitive performance much better than the more expensive and rigorous tests that we did on heavy metal levels, for example. So the actual strongest predictor of cognitive performance out of all 457 variables was mean corpuscular volume, a measure of red blood cell volume. This is indicative of chronic disease, so we measure this to look at microcytic anemia, which is great in terms of health equity because this is a blood test that people routinely get done in GP, and it's fairly cheap to perform. And if we can use this as a sort of measure to see if we can flag up who might be at risk of cognitive decline or cognitive deficit, then that's quite a straightforward test. So I included this just to show which measures were sort of positively and negatively correlated with cognitive performance. So as we'd sort of expect, an increase in C-reactive protein and white cell count, which are markers of systemic inflammation, corresponded with poorer cognition, whereas a measure of cell volume correlated positively with cognitive performance. Again, measures of hematocrit and hemoglobin, so oxygen carrying capacity measures, were negatively correlated with performance, interestingly. So looking further at the brain imaging data, this was the group where a lot more measures were associated with cognitive performance, which we kind of expect. Interestingly, the large regions like the corpus callosum and the cerebellum were predictive of cognitive performance, which is quite interesting because we normally look at basal ganglia regions, for example, the hippocampus we look at for memory and Alzheimer's disease, whereas actually the cerebellum and corpus callosum could actually be measured at much lower resolution and a lower field MRI scanner, which is much more cheaper and also more portable. We also found that some measures of brain iron, mainly the right amygdala iron, were predictive of cognitive performance as well, suggesting some interaction there. It wasn't just a smaller volume correlating with poorer cognitive performance. We actually found that swelling of some regions was associated with cognitive performance, which sort of fits with our molecular crowding hypothesis. Some things like the fourth ventricle volume you would expect to negatively correlate. So this is the space for CSF. So if the brain is atrophied, then that space is going to get bigger. So that is a marker of brain atrophy, but some regions like the entorhinal cortex, for example, were shown that swelling of these regions correlated with cognitive performance. And also again, amygdala iron, an increase in this iron was actually associated with poorer performance, which leads on to some theories we have, which I'll talk about later on in the presentation. Finally, the strongest predictive measures were actually atmospheric pollutant exposure. I included the time range that we had available for every year's data that we had available to us. Interestingly, for some, for example, lead, we saw that the year which was in between, back in the middle of the years that these people were assessed for brain and cognitive performance, that was the most predictive year. For example, with the carbon monoxide measures, these only went up to 2010, so the closest year to when they were measured was most predictive, which is what we would expect. The main predictors were measures associated with vehicle emissions and incineration, which suggests that some sort of local authority mapping and green corridors could aid in not only determining who's at high risk of poorer cognitive performance, but also in potentially being able to protect those people who are at a higher risk. Just to summarise what we've done in this cohort, we saw that expected risk factors like age and education and overcrowding predicted cognitive performance. In blood testing, we saw that routinely assessed markers of things like anemia and systemic inflammation were really strong predictors of cognitive performance. We saw that large regional volumes predicted cognitive performance, meaning that we have potential to look at a lower resolution or lower field. And we saw that measures of iron, including in the amygdala, were associated with this cognitive performance measure. In terms of environmental pollution, we saw that vehicle emission fumes and incineration associated atmospheric pollution measures were some of the strongest predictors of cognitive performance, suggesting that some local authority intervention would be good for this. So just to go back to one of my areas of interest, which is how the role of iron and heavy metals in cognitive performance and cognitive decline. I just wanted to highlight that some of the key predictors that that came out of the model were associated with iron processing and oxygen carrying capacity. And I'll talk a little bit more about this now, looking in a symptomatic and an at-risk cohort. So the symptomatic cohort we looked at had ALS, and in ALS we do see previous studies showing increased brain iron and also increased CSF levels of a marker for iron called ferritin, which is an iron storage molecule. And we think that potentially an increase in brain iron accumulation might be conferring neurodegeneration via specific mechanisms like ferroptosis, which is an iron-mediated form of cell death, and also localization to protein aggregates and amplification of this protein misfolding. To look at this, we looked further into this disease, which is a progressive degeneration of nerve cells. This disease occurs in around 5,000 adults in the UK at any given time. That's by far a rare disease, but it's thought to be caused by TDP43 aggregation in this localization, which is a protein involved in the disease. It can cause muscle weakness and stiffening, which can have a really wide range of symptoms, from motor symptoms to cognitive symptoms to gut symptoms. And again, like I said, we're seeing this increased brain iron in this disease, with previous studies showing a potential link between disease progression and increased brain iron. The TDP43, I'll just quickly introduce here. This is an RNA binding protein that's involved in RNA processing, and mislocalization and aggregation of this protein is observed in ALS. And there's some studies now showing that this protein is being seen in other diseases and in other contexts, for example, in Alzheimer's disease. And these disease-associated changes in TDP43 can impair its function, and the splicing of RNA playing a pathogenic role in the disease. We do have an antibody to look at this in disease cohorts, but we recently, in our lab, discovered or adapted a new tool to look at TDP43 where we can see earlier aggregation events or earlier pathological events. And so what we're looking for, I'm going to show you some images later on. So just briefly, what we're looking for in controls is this nice blue staining or a lack of brown, intense brown staining. And in this image, you can see the neuronal, so this is the nuclei of the cell, the cytoplasm, and you can see aggregates in the cytoplasm and also in the nucleus and the nucleolus of the cell that are not present in the controls. And we see a lot of different pathologies in these cells, so things like these rods in the nucleus and cytoplasmic punctate staining. So we looked in this cohort of sporadic ALS cases, which is a non-genetic form of ALS, which accounts for around 90% of ALS patients. And these patients all underwent cognitive testing using the Edinburgh Cognitive and Behavioural ALS screen. And we looked at post-mortem tissue from these patients in 6 different brain regions, where we looked at TDP43 pathology, so the characteristic of ALS disease, and we also stained for ferritin, which is the iron storage molecule or the marker for iron that we can look for in the brain. We actually found that amygdala TDP43 pathology was the most sensitive correlate of this behavioural dysfunction measured by the ECAS score. And you can see this in case 9, where someone had no behavioural deficits and they had quite weak TDP43 staining with these nice blue nuclei. Whereas in the case 4, who had behavioural deficit, you can see this really strong and intense brown staining with some rods in the nucleus and really intense cytoplasmic staining. Interestingly, when we then stained for ferritin, we found that ferritin levels not only correlated with the TDP43 pathology, both in and out of behavioural deficit, but ferritin alone was also able to distinguish between behavioural impairment in this cohort. I'll just take you quickly back to the asymptomatic cohort to show that while we saw that ferritin in the amygdala was able to distinguish between those with and without this cognitive impairment. In the asymptomatic cohort, right amygdala iron also came out as a key predictor of cognitive performance. And so we wanted to look for this pathology outside of ALS to see whether this link that we were seeing coming out in both of these cohorts could be to do with the presence of this pathology in people who didn't have ALS. So we looked at this cohort of people with ALS, people with Alzheimer's disease, and also non-disease controls from their 40s to their 80s, spanning across 4 different decades. We had around 10 people from each category where we looked at TDP43 staining in the postmortem tissue of the amygdala, hippocampus, and BA9 region, with the thought that the BA9 region is involved in ALS. The hippocampus is a region that would be involved potentially across diseases, including being the region that's involved heavily in Alzheimer's disease. And also we were seeing these associations in the amygdala, both in disease and outside of disease. We actually observed TDP43 pathology outside of ALS, with the key age-related change being in the amygdala TDP43 pathology, where we saw that while around 20% of people under 60 had TDP43 pathology in each of these brain regions, people who were over 60, who were these non-disease controls, actually around 75% of these people had TDP43 pathology. Another interesting thing to see here is that in the Alzheimer's disease patients, we see this really specific jump in seeing hippocampal TDP43 pathology, where even in the over 60s in the general population that we looked at, still only around 20% of people had this TDP43 pathology in the hippocampus, whereas in Alzheimer's disease, it jumps to around 70 to 80% of people having this pathology. So we've shown that measures of iron dysregulation are among the strongest predictors of cognitive performance, both in blood tests and also on brain scans. We showed that TDP43 pathology was correlated with ferritin in ALS patients, where ferritin was able to distinguish between those with and without this cognitive deficit. And we also showed that TDP43 pathology is present outside of ALS, both in controls and in other neurodegenerative diseases. Hopefully I've been able to show you that iron dysregulation in TDP43 pathology might represent interesting markers to look at for precision prevention strategies, irrespective of disease status. I'd just like to thank everyone in our lab, and also Gary Cameron, who performed the ICP-MS, Hamish Johnson, who we worked with to look at the pollution data, Olivia, who worked on the amygdala study, everyone from the Biomedical Imaging Centre for the help with the imaging work, and Alan and Shabina in the Rowett, who helped with the blood testing.